Hello class, you are once again welcome to AEM 722 Ruminant Animals. You remember in the last class we talked about ruminant animals and defined what they are and the examples we gave on them. In this class, we will be talking more on the digestive system of the ruminant animals. And for this class, I want you to pay attention so that we can discuss together as we go through the process. The course is AEM 722 Ruminant Animals, and our topic, which is the second topic of the week, is digestion in ruminant animals. I am your facilitator, Grace Johnson. As you can see on the screen, by way of introduction, in this lesson, you will be studying about the process of ruminant digestion. You will also be talking about functions of the various sections of the ruminant stomach. We will discuss the end products of microbial digestion in ruminants and how they are absorbed, as well as the anatomical differences between the young and the adult ruminants. And so, what is the digestion process in ruminants? As you can see from the diagram here, the stomach of the ruminants, like we have said earlier, is made up of 4 compartments. And it is these compartments that actually differentiate the ruminant animals from the conventional monogastric or non-ruminant animals. The rumen, as you can see in this picture, consists of pillars that have rhythmic contraction. And this contraction is actually what causes the contraction and relaxation of the rumen. And this helps in mixing the contents of the rumen. The rumen content is actually divided into 2 main parts. You have the bottom, which is the liquid phase, and you also have the top layer, which is a dense phase. And this is the floating phase. Solid materials or fibrous materials eaten by these animals are compacted in the dense floating part of the stomach. And it is this solid or the dense part that you normally see the animal regurgitating and eating from time to time as it appears to be resting. I'm sure you have all noticed that as you see sheep, goats, or cattle resting, you will see them chewing something. It is actually referred to as regurgitation, and it is this dense layer that they normally regurgitate back to the mouth and chew properly. The act of regurgitation actually helps in reducing particle size of these materials in the rumen and also increases surface area that bacteria can actually attach to and enhance the digestion process. How are carbohydrates digested in the ruminants? This is quite different from the monogastric animals. If you look at this diagram that we have here, it gives a diagrammatic representation of what occurs. And so food materials are consumed through the mouth that goes through the esophagus and it ends in the rumen. And there is now the back and forth movements, like we said, regurgitation of the food material from the rumen back to the mouth and back to the rumen until it is a paste is formed. In the rumen, there is major digestion take place, mostly microbial digestion. The rumen is actually a home to microorganisms such as bacteria, such as fungi, and they are actually, and protozoans, they are actually what digests fiber in the rumen. As it goes to the reticulum, there is a filtration where a lot of liquid materials is being absorbed. And when it gets to the omasum, there is the grinding of the further solid materials that is now left. The omasum is the same as the simple stomach of the non-ruminants, and that is where gastric digestion takes place, or otherwise referred to as enzymic digestion. And the food at this point of the abomasum is digested in the same way as food digested in man, in cats, and other non-ruminant animals. You then get to the duodenum and to the large intestine and small intestine, and food is voided out. So what you see here is actually the representation of what takes place. But the first part of it, if we may go back to our diagram, is the microbial attachment of food materials in the rumen. The fungi secrete enzymes that break down fiber, reducing the physical strength of food materials and allows bacteria and protozoa to be acted upon. The rumen is an anaerobic environment, that is to say it is oxygen-free, and therefore it is only microorganisms that can survive in the absence of oxygen that actually reside in the rumen. These microorganisms are able to break down the beta linkages in forages. Fibrous food materials are broken down into pyruvic acid, also referred to as the pyruvate, which is further oxidized to volatile fatty acids. Volatile fatty acids, also referred to as the VFAs, are actually the end product of microbial digestion in the rumen. The major fatty acids produced in the rumen are the acetic acid, the propionic acid, and the butyric acid. These are the major energy sources for ruminant animals. The VFA, or volatile fatty acids produced from the incomplete digestion of carbohydrates, is absorbed through the rumen wall. So the rumen serves two functions, both as a storage vat and as well as being an absorptive vat. So the lining of the wall of the rumen actually helps in absorption of food materials. Finally, the energy produced during the anaerobic fermentation is lower than what is produced under aerobic fermentation. Now, there is a difference between anaerobic and aerobic fermentation. The anaerobic, in the absence of oxygen, leads to the production of volatile fatty acids, like we have said. This is an incomplete digestion process, but the ruminant system can actually absorb this and utilize it as energy. But the energy efficiency is low. Under the normal non-ruminant digestion process, the end product of digestion is normally carbon dioxide and water. And this system that leads to the production of carbon dioxide and water in the abomasum that is also available with of oxygen is what brings about the end product of carbon dioxide and water, and it yields more energy. That is to say that the ruminant digestive system is not as efficient as the non-ruminant digestive system, where the production of energy is concerned. Energy or carbohydrate is one aspect of feed materials. The other aspect is the protein. How are proteins digested in the ruminants? Here again, I have a table for you to look at carefully. There are, when an animal eats feed materials, there are two forms of protein. There is the non-protein nitrogen, also referred to as the NPN. There is also the true proteins that are contained in the feed. These two types of proteins are digested differently. The true proteins that is contained in the feed, when eaten, gets to the rumen and is being degraded. There is also another form of the protein that is true protein, but it is referred to as rumen undegraded protein. That is to say, it bypasses the rumen and gets into the abomasum. That one, which goes through the rumen, is degraded by microorganisms. So irrespective of the quality of protein that is consumed, if it remains in the rumen, the microorganisms in the rumen will actually degrade this protein into microbial protein. And microbial protein could be lower than high quality protein. And so when you are feeding high quality protein to ruminants, it is better to present it in the form of undegraded protein so that it is not attacked by the microorganisms in the rumen, but goes directly into the abomasum and is the rumen undegraded protein. The microbial protein synthesized in the rumen by microorganisms is also utilized by the ruminant to produce protein. Now, the non-protein nitrogen could be in the form of urea that is being built up by the microorganisms. So that is to say that when low quality feed materials are eaten with low quality protein, it can be upgraded to microbial protein. And so the balancing act in terms of feeding of ruminants is very important so that you do not waste high quality protein or you allow the use of low quality protein to be improved upon by microorganisms. Let me go back a little. There is this diagram here that is you can look at and study more carefully. It's another presentation of this same process I have described here. Some proteins escape microbial fermentation in the rumen and pass unchanged into the abomasum. These are the things I want you to take home from the protein digestion in the rumen. And some of these proteins that escape are referred to as bypass proteins, rumen escape protein, rumen undegraded protein. These are various names, but they all refer to proteins that are not degraded in the rumen. The fraction of dietary proteins that is degraded in the rumen is referred to as the rumen degraded protein, also referred to as the RDP. The end product of digestion of rumen degraded protein are simple amino acids, ammonia, and urea. These are normally passed out through the eructation process in the animal. And some of the urea is also utilized, like we said, by microorganisms to synthesize proteins. The digestion leaves the reticulorumen through the reticulo-omasal orifice to the abomasum. So when the digestion process is completed, the digester leaves the reticulorumen through the reticulo-omasal orifice to the abomasum. And the major role of the abomasum, is the absorption of the huge volume of water that comes in with the animal. Now, that is what we have talked about in terms of the digestion of proteins in the rumen. Another aspect is what happens in the hind gut. After this has taken place, what happens behind the rumen, which is referred to as the hind gut? Once the digester enters the abomasum, it is subjected to a series of changes through the action of enzymes that is similar to what happens in the simple stomach of the monogastric animal. I have here a small table for you to look at that actually discusses the omasum, the role of the omasum and the abomasum within the process. When you have time, you can look at this table carefully and you'll be able to see the differences in terms of their performance in the ruminant animal. Now, there's an interesting thing for us to look at, the digestion in the young ruminant. One would have thought that once you are a ruminant, the digestive process is the same, but that's not true. For the young ruminant at birth, the rumen and reticulum together accounts for about 30% of the volume of the four chambers. You can see it in this diagram that is here. But by the age of 2 months, this would have increased to about 70%, And in the mature cow, the rumen and reticulum together occupies 85% of the volume of the stomach. This is to say that the development of the rumen is actually dependent on time and the nutrition of the animal. If you leave a mature ruminant solely on milk, then the rumen system will not be developed. And as the animal takes in milk, it passes straight into the abomasum. So it leaves the rumen undeveloped. But as the animal begins to grow and begins to eat solid fibrous materials, it brings about the initiation of microbial activity in the rumen. And the rumen now begins to develop and to grow. And at maturity, the rumen and the reticulum will now occupy 85% of the total size of the rumen. But as we have said earlier, at birth, the abomasum, which is the same as the true stomach of the non-ruminant, is actually what is the largest part. So this diagram gives you the development of the rumen from birth to adulthood of the ruminant animal. The abomasum, on the other hand, will have reduced from 70 to 7% of the volume of the rumen. In the young ruminants feeding solely on the dam's milk, the foregut digestion does not occur. Rather, milk is passed straight from the esophagus to the rumen through the esophageal groove. The esophageal groove is actually formed as a result of the reflex action of suckling. So once the animal begins to suckle, that reflex sends a signal to the system and the esophageal groove is formed. So let's just say another small tube is formed in the stomach that channels food. So as this action of suckling reduces, the reflex to create this groove now ceases, and the food now normally passes through the normal process in the adult stomach. This is an important aspect of the nutrition of a young calf on milk and is not subjected to microbial fermentation. So we should actually note that as a major difference between the digestive system of the young ruminant on milk and that of the adult ruminant on forage. Now, what are the advantages or disadvantages of the ruminant system? I would like to say that this fermentation that occurs has the potential to digest poor quality feeds and increase their feeding value. You know, we mentioned that the microorganisms will act on whatever material that is provided in the rumen. If it is of low quality, it can build it up because it converts it to microbial protein. On the other hand, if it is of high quality, it will still act on it and break it down and bring it to microbial protein. And so it has the ability to increase the value of feed materials if they are of low quality. But if they are of high quality, it can also reduce their nutritional value of these materials. Also, essential nutrients that are not found in the animal's diet, such as vitamins, amino acids, and fatty acids, are made by microbes and they are also used by the animal. So a lot of the B complex vitamins can be produced or synthesized by these microorganisms. And because the microorganisms reside in the rumen, they die and they are being absorbed in the rumen, these nutrients now become available to the animals. And therefore, the feeding of such nutrients is not too critical when you are considering ruminant animals. What are some of these disadvantages also? We have talked about the fact that fermentation has the potential to decrease the overall quality in high quality feeds, and that the fermentation process also requires and wastes a considerable amount of energy. We have made reference to the energy differential between the digestion that leads to the end product of carbon dioxide and water and the digestion that leads to volatile fatty acids as the end product. And that has effect in terms of the quality. of the energy that the animal eventually gets. So what have we learned in these units? Remember, this is unit 2, and in this lesson, that is lesson 2 of this class, we have been able to talk about the nature of the digestion process in the ruminants. We have also been able to talk about the difference between the fore and the hind gut digestion in ruminants. We have been able to talk about the end products of this digestion process, and also we have been able to talk about the differences between the digestion process of a young ruminant and an adult ruminant. Finally, we talked about the advantages and disadvantages of this digestion process. I thank you for having this good class, and I hope that you will study this video very well so that you will be able to understand further what we have done in this class. I'm sure that you have the PDF version in the virtual learning environment, and I also expect you to go through that to be able to complement what you have in this class. Thank you. Until we see again, I'm your facilitator, Grace Jafta.